Okay, good afternoon, everyone. My name is Steve Silverstein. I am now the former chair of the Melanoma Research Foundation after 8 years in that role, 12 years with the organization. And my background to get to this call today is I'm an 18-year stage 4 metastatic melanoma survivor. And back in the day, that was a big deal. Thank goodness today, many more people are getting through that. So it is an unbelievable privilege and honor for me to introduce you to two panelists on one of the most important subjects to patients, and I'd certainly come and bring that patient perspective. Um, Dr. Keith Flaherty and Ze'ev Ronai. Just a brief word about each, if I may. Keith is the Director of Clinical Research at Mass General, which is rated the #1 hospital in research hospital in the country. So you're getting the absolute best leading edge information. I met Keith personally 12 years ago or so when I first started getting involved with the MRF, and he had a vision for an organization to come together and create clinical trials that were not happening in the private sector. And that was a fascinating thing that the MRF got involved with. And so Keith began the education of myself and the like on clinical trials and the like. And Ze'ev Ronai is the head director of the Sanford Burnham Prebys NCI-Designated Cancer Center. Ze'ev tends more on the basic research side, and he's a brilliant scientist, and more importantly, he's got a critical eye for the whole community and bringing science together. And between the two of these gentlemen, you have literally the latest breaking news in the world on all cancer treatment. So today's subject is clinical trials, and it's a challenging subject. So I'm going to just ask a few questions to start. To Keith, could you please explain in very basic terms, what is a clinical trial and why would a patient want to consider taking on one?
Yeah, well, clinical trial it can mean a lot of things, but I think for today's discussion, we're going to be focusing mostly on treatment trials. Sometimes as are referred to as interventional trials. So like actually offering a therapy in the context of a trial. There's other types of trials that are focused on non-therapeutic either assessments of patients of their cancer, but kind of leave those aside for now. Maybe if we have time, we can come back to topics like that. But I think for treatment trials, which is typically the heart of what people are focused on when they think about clinical trials. Basically, the idea is that the clinical trials occupy a really broad space from the time that a potential therapy essentially leaves the laboratory and enters clinical investigation, in human beings for the very first time, referred to as phase 1 clinical trials, all the way through to the time of regulatory approval. So in the United States, that's FDA approval, and so that's a pretty broad range of experience and time. Also, in terms of numbers of patients will be treated, the number of years that transpire between phase 1 and FDA approval. But the point being that all across that course, the idea is to try to understand many things about a therapy, but fundamentally its efficacy, or how well it works, and its safety. And it's really the kind of relationship of those two things that constitutes, essentially what kind of net benefit we think a therapy offers, right? Because there's some therapies that confer very little risk, very little in the way of side effects, but they still have to balance that, even minimal risk with some evidence of benefit. Then there's therapies that are actually quite risky, and do actually pose real potential harm. But if the benefit is extraordinary, like let's say curative, right, for patients who are facing a life-threatening circumstance, then there are circumstances where the benefits can be demonstrated to clearly outweigh the risks, and that can lead to approval. But what I'm getting at here is that one has to satisfy kind of both sides of that equation, if you will, understanding the benefit, understanding the risk. And that needs to be done to a sufficient degree to satisfy, in the US environment, the FDA, the Food and Drug Administration, that we know enough, right, about both the potential benefits and the potential risks for this to then become a widely available treatment. So that's kind of, I think, the main thrust of what we'll probably dive into in today's discussion. I will mention, just as a last comment, that there are clinical trials that are conducted sometimes comparing therapies that have already been through the whole process that I just described, right? In other words, where now you've got, let's say, 2 or more treatments that are of known benefit, but we don't know which one is better than the other, right? And then there can be clinical trials that will actually, do those types of direct comparisons. So, that's, I think, gives you a broad sketch of really what is a very wide range of types of clinical trials, and we can delve into that further.
So if I can follow up on that, it is such a broad range, and I've always said over time, the worst person to make a decision about your medical care is the patient, because we're least qualified. But how do you begin to think about advising a patient to go either into a clinical trial first, or take, and today in the melanoma space, we have wonderful drugs that get 50-60% of the patients through. Can you explain what that process is to help a patient decide?
Yeah, sure. Well, actually, let me wind back the clock a little bit, to 2010 and before we had these treatments, right? Because back then, there were such things as melanoma therapies, but they really had very unreliable efficacy, which is to say that ballpark, let's say 10% of patients could derive some benefit, and there was some significant risks associated with those therapies also. And so a low likelihood of benefit and with some risk as well, that landscape of available treatment options, again, about 10 years ago and before, were considered to be sufficiently uncompelling that it was reasonable to contemplate participating in a clinical trial right away, essentially skipping over those therapies, right, going straight into a clinical trial. And what I mean here is, like, even testing a therapy that was very new and very unproven, right? That didn't have a track record yet. Now, in many cancer types still today, we still face that problem. Like the available therapies that do exist simply don't have sufficient track record to make them, very compelling in the eyes of patients and their doctors. In which case, participating in a clinical trial as a first consideration is still highly relevant. In melanoma, as you say, we've now, let's say for the past 10 years, we've had quite compelling therapies that have made it through clinical trials, made it through FDA approval. And depending on some of the specifics in terms of the type of melanoma and even its molecular makeup, that menu of options is, of variable length. But the point being that we have therapies that we know that for patients, for example, who have metastatic melanoma, they clearly extend survival. And can even potentially cure a subpopulation or minority of patients. In the post-surgical setting where we're worried about the possibility there might be residual microscopic disease, those same therapies can clearly eradicate that and cure beyond what surgery can do, evidence of melanoma. So in either situation, you'd say, well, those are pretty compelling therapies, the new treatments that we have for melanoma. And those have to be discussed in detail first, right? So, patient has to understand those options before then considering a clinical trial. But it's not necessarily an either or decision. And that's maybe a piece just to kind of flag as we kind of keep, dilating into kind of the details of what constitutes a clinical trial. So, for example, in melanoma, the most common type of clinical trial will be investigating a new therapy on top of one of those existing therapies, right? So like a combination approach where you've got, if you will, the backbone of the FDA approved treatment. And in addition to that, another therapy is being added. And so whether that's the right option or not, right, when someone's considering their options, well, it's all a matter of the details, right? Which is to say, okay, you've got the standard of care component maybe embedded in such a trial, but then your focus needs to attend to, well, what do we know about the new agent, right? What are the potential benefits and what are the potential risks? And emphasize the word potential, right? Because until such time as the therapy receives regulatory approval and becomes an approved standard, everyone has to understand that we don't fully understand the benefits and the risks, right? We have a kind of a provisional understanding that gets better and better and better as more patients are treated, as more time transpires. But it is a gradual process, and that's what we spend a lot of time in discussion as we first really focus on the available therapies. What are their upsides and downsides, and then kind of shift to what are the emerging new potential treatments that could be considered even as an add-on in terms of upfront treatment decision making.
So, well, when we're on this, if I may, why would a patient help guide that decision or be part of that decision? Or is that mostly meant for the physician to say, this is the treatment, but I really recommend this for you? How does that dynamic play out?
Well, this is a constant issue in the practice of medicine in terms of really wanting, always to try to have, patient and their care providers understand, essentially what they're up against in terms of the underlying condition, right? So like not all cancers are the same, and not all, even within a cancer type like melanoma, not all considerations of treatment or treatment settings are the same. So before I mentioned, having metastatic disease, right? So, beyond what surgery can manage, right? Beyond what, kind of local therapy can potentially remove or eradicate. So when you're beyond the reach of, a surgical approach, we, quickly start moving into medical therapy discussions, understanding that you're in that situation and the potential, threats that the underlying disease poses, right? Versus the post-operative setting where all evidence of a cancer has been removed, but there might be a chance, let's say a 10% chance or 30% chance, 50% chance, 80% chance that there actually might still be microscopic disease that we can't see on a scan, right? But where at increasing levels of concern, one might be willing to contemplate, drug therapy. What I'm getting at here is you have to understand, the fundamental situation you're in. So that's kind of the point of discussion and education that comes first and foremost, and then start talking about the treatments, right? And so what I'm getting at here is that, okay, we might have an available therapy that benefits 20% of patients. I mean, just use that as a number. We're grateful actually that we now have in melanoma even more effective treatments than that. And as you describe what that 20% means, what do we mean by benefit? There are some people who hear 20% and say, well, that's quite a bit greater than zero, right? And there's others who say, well, that's quite a bit short of 100%, right? And I'm facing, let's say, a life-threatening circumstance, right? I would like to try to, I would like to discuss or explore, those options that might exist that would, kind of help potentially offer more than 20%, right? I mean, where I want some upside. Now, if you're talking about a clinical trial, formally speaking, that means we don't exactly know what the new number might be, right? The new treatment, let's say, added to a standard therapy. Does it get you to 30%? Does it get you to 50% benefit or likelihood of benefit? We oftentimes, again, can offer some estimate based on what information we have, number of patients who've been treated, right? And like this, from early trials, that's a small number. From later trials, it's a larger number. And so this is, again, a matter of trying to help people understand what we know and what we don't know. But it's really, what I'm trying to suggest is there's just no black and white here, right? There's no walking in the room and saying, I've just met you for the very first time. This is, the best option for you. And I can say that with some degree of certainty, regarding a therapy that's still in clinical trials. Like, no. I mean, even when talking about available treatments outside of clinical trials, it's a matter of education and really level setting in terms of one situation, what the available treatments offer, and then what might be the potential upside based on current evidence from so-called investigational therapies.
But if I may, I think technological advance that we encounter or benefit from over the last few years allow us to make much more educated decision as per what a patient may benefit, or if there are those 20%, how to stratify those 20% to treatment. We have now new tools. Our toolbox has changed, and we have way better accuracy in terms of figuring out what may a patient benefit from and what may not. And based on that, we are able to drive clinical trials in a greater success. So the whole formula has changed just in recent years, thanks to the technology.
No, that's right. I mean, and I would say, broadly construed, that's, sort of diagnostic technology, right? So like we make a diagnosis of a type of cancer, but these days, and certainly in melanoma, we go beyond that, and we start applying molecular diagnostics and really trying to understand, exactly what drove the formation of this, cancer in the first place, and what might be its therapeutic vulnerabilities. Now that in every cancer type, but let's, again, use melanoma here as the example. that's while we've made some real strides and we have some approved tests that are used all day, every day in clinical practice, that is another area where there continue to be new advances, new discoveries, and where we're on the diagnostic side trying to understand, in the same way that we're trying to understand about therapies, kind of the benefits they offer, to what degree do new diagnostics better allow us to understand, okay, your chance of responding to this therapy is not 20%, it's 60%, because you've got particular molecular makeup, if you will. And unfortunately, there on the other side of that, there can be patients who their molecular diagnosis, if you will, might mean that they have a very, very small chance of benefiting from that therapy. But that's critical, back to one of your original questions, Steve, because if you're in that circumstance where available therapy offers you very, very little, you may have to really be thinking about pushing ahead to novel treatments in clinical trials, essentially skipping over these, yes, available treatments, but ones that would potentially offer you very little benefit. So this stratification notion is really, really important. And again, it's another component of this, rather long conversation or set of conversations that we have with every patient as they're getting oriented to their diagnosis and their options.
You bring up a fascinating point. The prioritization of how you select your treatments, because if you do certain things, it might disqualify you from other issues. How does the community oncologist stay aware of this? I mean, my many years now in this community tells me you need to go to the best centers to get the latest breaking, because it's just changing so rapidly. So how, is there any thought on at least go back to the prioritization? How does a patient begin to wrap their head around that?
No, that's right. I mean, this is, certainly a complicated issue. And you're right that as soon as you go beyond the question of, okay, I've just been diagnosed, what's my best, first treatment, if you will, when you start saying, well, what's my best backup option? And how do I think about a first treatment as it relates to my backup option? This is certainly in melanoma, where we've had, essentially a flood of new therapies. This becomes a really important issue. And once you get to that level of complexity, it's true that increasingly it is very helpful to have the opinion of someone who just thinks about the cancer type or even the molecular type of cancer that you have. They may not be the person that needs to supervise your actual treatment, right? But just conferring with such a person can be quite helpful. Increasingly these days, thankfully, that can happen kind of remotely or digitally, if you will, without having to travel around necessarily. But no, this is an important point before getting to clinical trials. I mean, right. So if you've got multiple available options, that are approved and, kind of could be prescribed, one does, increasingly these days need to think about, kind of at least as far as, first approach and backup approach, and what makes the most sense as a, if you will, 1-2 punch. There can be circumstances, and I think this might be behind your question as well, where clinical trials, are more available or less available based on the treatments that you've already pursued. Now, in general, that's not a constraint. If you're pursuing a therapy that is of known clear benefit, I mean, it extends survival time for patients who have metastatic cancer, or increases cure rate after surgery in the so-called adjuvant setting. Generally speaking, clinical trials will not prohibit participation or access, if you will, based on that. But there can be other therapies that, let's say, are of more marginal benefit that exist. They're out there, they're available, where pursuing those actually could block the ability to participate in a clinical trial. It's offering a treatment that's showing more and more early success, let's say, through the course of dozens and then hundreds of patients treated. So this becomes really important, and I think just further emphasizes your point that at some point in the discussion, and certainly if someone has tried a treatment and it has proven not to be effective, and now they're looking for a next treatment, kind of conferring with an expert who has an eye towards emerging investigational therapies that might be 6 years into clinical trials, hundreds of patients treated, where we're actually we now know a fair amount, and it's showing real promise and looks like it actually is likely heading towards eventual FDA approval. Drugs that or therapies that enter that zone, people kind of need to know about, because it does start to inform treatment decision making, versus what might remain in the available treatment armamentarium.
Yes, you were among the first in the country, maybe the first in the country, that monitored the response to clinical trial in real time, which means samples were collected from patients prior, during, after. And that was a humongous resource to figure out, well, what are the molecular marker changes and what is the success of the trial? So technology helped us to reach to a point where we can figure out what's going on in real time. And how you look at that and how this will dictate where we go now into the future.
Yeah, well, I would say that up until now, all of that work of trying to understand, what I might summarize as sort of molecular response, is a drug doing its job? Like, is it actually doing what we, thought it could do based on laboratory research? That largely is the type of investigation that, was its own kind of research, right? So the treatment might be offered in a clinical trial aiming to offer benefit to a patient, but the understanding of the, molecular, function, if you will, is more to try to better understand that therapy to, advance treatment for the next wave of patients, if you will, that come along and are, facing a decision about, now this same treatment versus yet another, treatment that's now emerging. But that's beginning to change. In other words, for that to be purely to try to inform, what it is that we pursue, a few years down the line in terms of, next approaches. Now, increasingly, there are these molecular response tools that are now entering clinical practice. Many of them have to do with, simply trying to improve upon scans. So CAT scans, MRIs, what we've conventionally used to monitor treatment response. We now have blood-based markers that are, frankly, can tell us a lot earlier in the course of treatment. And those can be somewhat customized, actually, to not just cancer type, but even molecular type. And so you're absolutely right that what, was, 5 to 10 years ago, research in that realm has now translated into clinical practice, where we, get closer and closer to understanding whether the drug is actually doing its job, not after 2 months or 3 months of treatment, but, within a week of treatment. That's, that is actually now emerging as a new standard approach. So, this is a whole nother moving part in clinical trials, is this issue of trying to understand, what I'm referring to as molecular response. And we're continually working on this area because, to the point that Steve and you raised before, we would like to have a better and better crystal ball to really be able to take, let's say, a therapy that works in 50% of the population, right, and say, okay, who are the 50% who benefit, right? They should get it, because in that population, that's 100% benefit. And then who are the patients for whom we simply need to do better? We need to try, totally new things. So that's stratification, the term you used before. But then you start a treatment, right? And it's got, let's go back to 50-50, 50% chance of working, 50% chance of not working. You'd like to know that as soon as possible, right? Within days to a week, let's say, based on, some current technologies, so that then you could pivot and move to a second therapy. Back to Steve's point, if you've already mapped out what your second approach would be, well, move quickly to that second approach. And again, that was a dream 5 and 10 years ago, that we actually could maneuver that quickly. But it's I would say in 2021, it's already become a reality, at least in certain cancer types.
Yeah. So just follow that up, where, and it kind of goes to the work that Ze'ev's doing in his thing, that all this molecular work that I had the pleasure of touring the facility last week, and to see the equipment and the science being done at this level is remarkable. So it sounds, and it's news to me, it's fascinating that we can now start to direct, as opposed to, okay, you look like a good candidate for this clinical trial 15 years ago. Now it's like, wait, we did the blood work, you're not a good, or you are a perfect candidate. So 3 to 5 years, from now, how do you see this ability to really steer into clinical trials or any other treatment protocol?
Yeah, no, that's right. Yeah, I mean, I would combine the kind of last two discussion points into one. I mean, really having a much stronger precision medicine focus, right? So really using molecular diagnostics to inform treatment, triaging, or choosing between available therapies, investigational therapies, or the add-on of investigational therapies, as we discussed before. Basically taking what can be a very complicated, map, and making it much more clear, right? To say, well, no, like for you, it's only this, it's these few options that we need to focus on, not 20 options, not if you go to clinicaltrials.gov,
Right.
the
It's overwhelming.
but it's overwhelming. You put in a
Overwhelming.
diagnosis of melanoma and you come up with 473 hits, but this is where molecular diagnostics go a long way towards reducing that down to the appropriate and manageable number. But then again, layer on top of that, what we just discussed in terms of molecular response monitoring, where again you're making smarter decisions up front and you're confirming that you made the right decision. Not months later, not waiting to see if the cancer gets worse. No, like understand very early on, and then move as needed to backup options. So I think I see the combining of those two to be well within view in the 5-year horizon. But maybe just add one more point, which is that the ability to detect cancer that is beyond the reach of what scans can pick up. I mentioned this concept before. What we call adjuvant therapy is offered to people who have surgery to remove their cancer, but where we have concern based on historical data that people in that circumstance will have their cancer return over subsequent months or years based on microscopic disease having been left behind, where we can't see, right? But finding that using blood-based diagnostic technologies, it is now entering clinical practice as we speak. I mean, these highly sensitive blood-based methods for picking up the molecular fingerprint, if you will, and this has been research of 7-8 years duration that's now hitting clinical practice, where we can now think about taking the same therapies that we used to hold off and only use when there was evidence that the cancer had come back, right, where we were 100% certain at that point. Now moving those same therapies into the situation where all we can see are traces of it in the blood, right? We're optimistic, but we haven't proven yet that those same therapies are actually going to be vastly more effective because we're now directing it against many fewer cancer cells, right? So now we're like taking the same therapies that could kind of hold back the disease and buy time and actually translate those into truly curative therapies in that setting. This is, I think, the big push of the next 5 years, wedding together the diagnostics and the therapeutics.
So are there many clinical trials on adjuvant therapy? Because it was one of the topics I had that I think that this population listening to, the folks listening today, need to be very much made aware of. You just explained it nicely. Are there many studies out right now for that?
Oh, for sure. But let me just clarify a little bit that term of that use, adjuvant. So we've used the term adjuvant, and many patients, if they've gone through cancer diagnosis and discussion, they'll have heard that term. We used that term historically to mean we did everything we can with surgery. We've done scans to look for evidence of cancer elsewhere, and we don't see it. But, right, based on 10 years ago, 20 years ago experience with 1,000 people, 10,000 people with the same diagnosis, we know that, I'm just picking a number here, half those patients had their cancer return with the same, basic diagnosis, the same features, right? Half did, half don't, right? But you're diagnosed with that problem right here and right now. You want to know, do I have the problem or do I not, right? So it up until recent time, as in literally this almost up to this discussion practically, we would say we don't know. We don't know with if you're in the 50% group who still got the problem or not, and only time and more scans typically will tell us. And so using this blood-based methodology, what I was alluding to there before, now we're actually going to be able to nail down much more accurately who's still got the problem, right? That scans weren't able to show. If we give treatment, then calling that adjuvant isn't quite right, because the term adjuvant meant we don't know, right? We're kind of blind to whether the problem really is there, really is not there, but we're going to offer the treatment on the out of the concern that it is there. So I would call that something different than adjuvant, right? It's essentially a new cancer stage. I mean, it's finding a cancer in blood. Here I'm talking about what we call solid tumors. So whether it's melanoma, skin cancer, or lung cancer, or colon cancer, finding the traces of it in the blood and directing therapy there. There's an explosion in the number of clinical trials that are directed by that methodology. But that was not true, 2 and 3 years ago. Concept. It wasn't sufficiently well developed in the technology to even launch a clinical trial in that direction.
So it's fascinating. What would you call this, and how do we get the patient population aware that this is out there? Because a lot of people, oh, I have my surgery, I'm clean, I'm done. You
Yeah.
know, and they don't necessarily hear that, right?
Yeah, there's still not a consensus. There's still not a consensus term for this. But what I use the term blood-based biomarker positive or persistently positive. The common technology, which I haven't mentioned yet, is circulating tumor DNA. Right. So the DNA that comes from the tumor cells being shed into the bloodstream, and so doing such a test before surgery, fine, but most importantly after surgery, weeks after surgery, and seeing that it's persistently positive tells you that you've got someone in front of you who basically still has the problem. And so a persistently circulating tumor DNA positive population of patients, this is the jargon term that we use here. This new diagnostic setting is what's motivating these clinical trials. There's a number underway as we speak, but it's going to multiply tenfold over even just this coming year, because the diagnostic tools have been sufficiently well developed to actually use them to basically inform patients and their doctors, look, you've got a problem, and we've got a clinical trial that will offer you therapy to address that problem. And it could be a readily available therapy that already exists, but that, as I said before, we would otherwise, reserve until there was 100% certainty that there's, a cancer diagnosis, as in on scans at some subsequent time.
So if I could just change the subject a tiny bit more, there are all these different trials going on. I've heard in the past, we're having trouble accruing patients to them. There's so many different ones. And maybe if you could speed up that process, we can get through these trials faster. So could you comment on that methodology of accruing patients and closing studies and moving forward? Yeah,
well, let me just first quickly, highlight the fact that in the United States, anyway, about 10% of cancer patients participate in clinical trials. So that's a large fraction who don't, and it's largely because they're not accessible to many, many patients. Which is to say that those clinical trials are not available at the in the clinical practice environment where they're being treated, right? And so that's a problem. And as a field, we've been doing more and more, really just within the past, I would say 5 years, to really try to change that by streamlining clinical trials and making it possible for them to be conducted in a broader range of settings. I want to take this molecular diagnostic concept that we touched on before, right, where there are certain new therapies that are exquisitely paired with a specific molecular and often genetic alteration that might only be found in a small percentage of all cancer patients. And increasingly, you find emerging therapies that are being developed for that population of patients who might be, quote unquote, hard to find, where the trials are really going to the patient, right? And where it's not a matter of having the patient have to find the big medical center that's that might be hundreds, thousands of miles away even, to enroll in a clinical trial, but actually flip that and actually bring the clinical trial literally right down the road to the practice environment where the patient's being treated. That has increasingly been happening, but solving that is a really critical issue. What I would say in the meantime, though, the single biggest problem, and one that I and others have been really trying to wrestle down for a number of years, is simply trying to make it crystal clear what the trial options are that are most relevant, right? And that's not 472 clinical trials, just repeating the number that I mentioned before, right? It's to make it crystal clear that someone who's just been confronted with their diagnosis, and they now have a molecular diagnosis beyond that, what are the top, just pick a manageable number that we can all wrap our minds around. 3 options. Does that include a clinical trial? Yes or no, right? I mean, it's like that shouldn't be a patient's job to sort that out, right? It's weaving these things together, what we refer to as decision support for care providers around molecular diagnostics. This has been a huge unmet need in the field, to just make it crystal clear to doctors who can then make it crystal clear to their patients. Look, this is the rank order list, and where a clinical trial falls in that list, sometimes it's number 1, sometimes it's number 2, all depends on the circumstance. But the point being that whether pursuing a clinical trial is important, whether it's the very best option to pursue, right? That needs to be demystified in the same way that we focus on just wrestling down the available therapy options. And I would say we, as a field, have done quite a poor job of making that very, very transparent. Until we do that, I would say it's quite understandable to me why the vast majority of cancer patients in the United States don't participate in clinical trial, because it's just, the crystal ball is not at all clear as to like what produces the best possible outcome for me.
So interesting. You're creating massive data, and it sounds like this is ripe for AI type of screening, filtering methodologies. Is that happening in the field? Are you seeing
It
that
is.
discipline?
It is.
I mean,
Yeah.
Ze'ev's always been a great proponent of let's bring multiple disciplines together and figure this out. Now it seems like it's really prime and ready for that type of.
It is, but it's early days and kind of frustrating in a way. I'll tell you that the flow of data that you're referring to is not as free flowing as we would like, to be totally clear. There's a variety of reasons for that, but that is frustrating to witness, because you could argue that over the past decade, the advances in therapy, our molecular diagnostic progress as well, if you just look back and ask for an accounting of, well, how did the American cancer population do, in this past 10 years, down to the individual patient level, so really understand, if you've got this specific type of melanoma with these molecular features, this is how, based on 10 years worth of, past experience, this is how the available treatments performed, right? So the chemotherapy drugs that were developed 50 years ago, this is how, those therapies, performed. This is how the new emerging immunotherapy or the new molecularly targeted therapy performed, right? So that you actually had, real data to drive this discussion, right? And understand, as I said before, what the situation is that you're facing, like in a very granular way. What is the rank order of best available treatments? I mean, I still have conversations with patients today that are driven by controlled clinical trials, right? These like trials that enrolled, a few hundred patients, where the requirements, the stringency of those trials was such that, that the patient in front of me may not actually be a great match or a great fit to that population. And in the meantime, 10,000, 20,000 patients have been treated since that clinical trial was done, right? And I and the patient would like to have the conversation be driven by the reality of, how does this therapy perform at the 20,000 patient level, so that now we can really understand between available treatment options and an emerging clinical trial option, which is the best, as opposed to this, at best semi-quantitative notion. So this will improve. I mean, it is improving. The software, the technology, the AI, that's not the rate limiting piece right now. We literally just need the flow of data into such tools to be able to, I think, better inform this landscape of decision making.
Would you agree that it is easier for us to reach a point of clinical trial today? So from scientific perspective, making a discovery and driving an investigator-initiated clinical trial could be much faster than it was 5-10 years ago. And similarly, a company, a pharma that has a new drug would like to put it to test, they have much easier life, or the route is shorter. Do you agree with that?
Well, certainly that's true if the discovery has to do with who should receive that therapy. In other words, you get back to the molecular diagnostics. That's right. So I mean, oftentimes these days, a new therapy will enter clinical trials, the very first clinical trials, with some notion of which population is potentially most likely to benefit. But inevitably, there'll be discoveries made, in parallel, essentially, right? The drug's entering clinical trials, and discoveries are made regarding, new molecular markers, molecular features that, in the laboratory anyway, seem to associate with potential responsiveness, and the ability to add those in or direct the direction in which a therapy is being developed. That is absolutely, I mean, compared to 5 and certainly 10 years ago, is just a night and day difference to be able to respond to that type of information. In terms of the threshold to get a new therapy into clinical trials, I would say that's, yes, it's the process, the science of that has improved. I'd say that's a fair statement. That said, there is a fundamental principle in terms of having to understand a certain amount of safety information before a therapy ever reaches a human being. So, this is, in so-called preclinical, experiments, certain threshold there that remains at the same level it did 10 years ago. But pairing that with the safety, the efficacy insights, I would say that's where the biggest improvement has been. The ability to be able to evaluate therapies before they ever go into patients, to really understand which ones have promise, and streamline their entry into clinical trials, matching them with the right patients. That's where the improvements have been seen. So it's still a pretty high threshold or high bar to demonstrate that there's sufficient safety to go into human investigation. But the efficacy and the molecular diagnostic side, that's where the big advances have been.
We went a long way from chemotherapy to targeted therapy and now to immunotherapy. And not only that, but we do that now in combination.
Yeah.
So the level of sophistication in driving those clinical trials is incomparable to what we have used to do in the past. Immunotherapy today is 50% or more of what we usually do, especially in melanoma. How we make those decisions? What drives?
So that I would say that's where, if you were to divide the world of new therapies into so-called targeted therapies, which were usually means that the therapy, all drugs travel around the body, in a fairly widespread way. But targeted therapies are generally designed to enter into cells, not only cancer cells, unfortunately, that we don't have that, we're not that smart, but all cells, but where the vulnerability to those drugs is in cancer cells, principally. We refer to those as targeted therapies, versus immunotherapy, which is trying to motivate the immune system to attack the cancer. So that's kind of indirect, right? It's not those drugs aren't directly doing their work on the cancer cell. I would say that the pairing of molecular diagnostics with targeted therapy, that has been a pretty tight relationship. The same precision medicine principles have not been brought to bear on the immunotherapy side. And yet now when we look back over the past 10 years in melanoma, for example, lots of data that's been published on the topic, there is molecular information that allows you to say with at least 90% confidence, I know who's going to respond or who's not going to respond to this available immunotherapy, which is far better than the kind of 40-60 split that we face right now. And so this is where I would say that we're still catching up in the immunotherapy realm to have the same sort of standard applied, where we take this molecular information, translate it into clinical practice so that it's part of our decision making between the available therapies as well as for clinical trial participation. And your point about combinations is very well put. Here we are in melanoma and many other cancers having found kind of new foundations, if you will, upon which to build. I mean, truly effective therapies, but not beneficial for everybody. But they are foundations upon which to build. And there is this crosstalk between targeted therapy and immunotherapy that's been fairly heavily explored in clinical trials. But this is only going to get more complicated in the years ahead, because so many of the discoveries that are being made are regarding what I would refer to as secondary vulnerabilities, right? In other words, like therapies that by themselves don't actually do a lot, but when combined with one of these foundational therapies can do an enormous amount. And so scaling up to these sort of higher order combination treatments, that's the work of the next 5 and 10 years across cancer broadly, but certainly in melanoma. And again, just makes the conversation a little bit more complicated in terms of understanding, well, how do you use molecular insights to understand a 3-drug combination, and boil all this down to the individual patient in terms of this is the right fit for you. This is where we're going. I mean, just to be totally clear as a field, but there's multiple moving parts, and it does require some expertise in terms of kind of talking through and thinking through, that level of decision making.
Steve?
Fascinating discussion. It's just remarkable where the field has come in 18 years in my participation in it, and to see how quickly now you're really getting a lot of speed, and the cross-pollinating between the different disciplines is really wonderful to see. So there have been just a lot of talk about side effects, and I know a lot of patients are concerned about that, especially in clinical trials. Can you give us an insight there where we were a few years back where it was fairly rough? Or have things gotten more refined, or is that still a risk there?
No. Well, certainly a risk, but that's, again, a lot of advance has been made in terms of being able to predict the side effect profile of therapies before the first human has ever even treated, right? And so this is true with so-called targeted therapies, as we talked about before. Similarly, increasingly true with immunotherapies. And so, I would say there's much less guesswork, if you will, in terms of understanding what side effect risks there might be. But as I mentioned before, when talking about clinical trials, you need to know where you are in the sequence of patients in the world, treated with this therapy. You're entitled to know not just, that I'm the 47th or the 547th patient to receive this therapy, but what has been the experience up until now, right? In terms of, side effects that have been observed. Because that, of course, is a very different discussion depending on how much is known, how relatively little might be known. But certainly, again, it's this ability to be able to look back over now decades worth of therapies developed, these more precise medicines that pose less risk, where I would say it's a lot less intimidating, frankly, as a doctor talking to patients, to be able to sort of describe for them, like, look, here's the potential downsides. For many of these targeted therapies, they're pill-based treatments where side effects might occur, but they're gradual in onset. And as long as we have an open line of communication, we can just tell you, stop taking the pills, take a break, right? Side effects will dissipate and we'll decide what to do, once they do, in terms of restarting or adjusting the dose. With intravenous drugs, it can be a little bit more challenging because once they're in, a little hard to remove them, but nonetheless, I would say there also, we do have monitoring approaches that allow us to keep, to ensure that patients are safer, than in the years past where we didn't have those tools. So that's its own important part of the conversation, right? I mean, this is just absolutely, I spend as much time talking about the potential upsides in terms of, efficacy or benefit as I do on the risk side. Even for therapies where the benefits vastly outweigh the risks, right? You still have to really make crystal clear on both sides. And that's true for FDA approved therapies, the same as for clinical trial drugs.
Yeah, it's a remarkable conversation, and again, many years ago, you would be thrilled just to have an opportunity, if you were at that extreme end of the case as a patient, to have that and deal with the side effects. But to hear that we've been able to deal with them so much more sophisticated now, it's really heartwarming to hear. Ze'ev, other thoughts on your?
I think that we really went a long way, and we have so many options today, which we didn't have in the past. And we understand how to put them together in a better way than ever before. So it looks good to the future. Would you agree, Kate? You're taking care of the patients.
No doubt. No, I think the marriage of science and medicine in the past, 20 years, you just look at the kind of the rate of progress, molecular understanding of cancer, translating that into therapy. It's really gotten, to this point, quite steep. I mean, the pace of progress is really, every year, more progress made, multiple new, larger number of new therapies advanced, that show real benefit, and escalating numbers of therapies approved. So, it's hard not to be optimistic, that as technology has continued to advance, we're going to keep seeing, an upslope in that pace of progress. But we have a lot of work to do, and it is this marriage of sort of science and medicine, not in this kind of pass-off way, right? Where the science is done in the laboratory and then the clinical trials are done, to kind of establish is this a medicine or not? But really weaving the two together, as I think we've hammered on multiple times here, keeping the science going, if you will, through clinical trials, after drugs approved even. To continue refining their application. That, again, that was not a thing, in the decade of the 2000s. Became a thing in the 2010s. That's going to be the thing, from here on out. So, I'm certainly optimistic that as we really, kind of weave these points, that we've covered, more tightly together, that's going to accelerate progress for patients.
Hey, Ze'ev, how do you see that from your perspective and your team? I mean, is it also rapidly starting to build up now, the breakthroughs and the speed to, explore each of these different channels?
You're asking me?
Yeah, I'm sorry,
Oh,
Ze'ev.
yeah,
Yeah.
sure.
Yeah.
Sure. I think that the one thing that we absolutely benefit is technology, and the technological advances that presented to us allow us to look at tumors in a way that we have never been able to see before. Resolution, single cell, understanding the heterogeneity that allow us to tease out why tumor are resistant, why tumor is more metastatic, and so on and so forth. And it allows us to work with the clinicians like Keith and analyze samples from patients upon treatment and figure out what happened, why a patient stopped responding, why a patient did respond. And then that paves a major roads into how we do, what are we going to do next, and what's going to be a better approach, or what are the next molecular markers that we need to pay attention to. So, and largely it's technology, computer-based, but not only, that redefine the path and dictate our future. And it's really exciting.
Certainly is. Okay. Any other questions or comments you'd like to explore today, Ze'ev?
No, I think that you've done a remarkable job, Steve and Keith, together. I really appreciate it. I think that the audience have enjoyed learning as much as I did. And thank you so much for the time. Wonderful.
Yeah, pleasure.
Thanks.
Thank you very much.
Great. Thanks.